Hello, everyone. All right, I'd like to thank the Nestle for having me and all the chairs for inviting me here to talk today. As Dr. Walker said, my talk is a little bit different. We've been talking a lot about the bacteria, and so today I'm going to talk about the viruses, or the virome. Okay, so, and if that blank slide of what we don't know about the milk, the virome has even less data. Okay, so viruses actually comprise the highest number of species on Earth, or highest number of different particles, so 10 to the 31 different viral particles exist on Earth. And in just 1 ml of seawater, you can find 10 to the 7th viruses. That's really a crazy number if you just think about that. And so viruses, of course, because they're everywhere on Earth, we are going to find them also on the human body. So today I'll talk a bit about the virome of human milk and infant gut, the very few studies that exist out there. Talk about virome transmission with the focus on mother to infant, and then describe the role of the virome on infant health. And so when we think about the infant gut, this is the gut here, we've been mostly concentrating on the bacteria. But in addition to the bacteria, we have these eukaryotic viruses that live there, and then the bacteriophages. And they all play very important roles. So most of the time when you think about human viruses, you think about transient viral infections. These are things like rhinovirus or coronavirus that cause the common cold. There's lots of these viruses out there. And then there are the persistent or latent viruses. These are in the herpes family, for example, that can cause cold sores that they persist in your body and come back every time your immune system is a little bit depressed. And then there's endogenous retroviruses, and these actually make up about 8% of the human DNA. And they are incorporated into the human DNA, and then actually can even be incorporated into the germ cells and get passed from generation to the next generation. And then there are the bacteriophages. These are viruses that infect bacteria, and they comprise they are about 10 to 11 bacteriophages per gram of stool. And then, in addition to all of that, the majority of the viruses that we identify when we study the virome are actually novel, unidentified viruses that we have no idea about yet. So here are some pictures of bacteriophages. And these are bacteriophages that are attached to a bacteria. You could see there's just so many of them here, and again here in yellow, attached to this bacterial cell. And so why do we care about these bacteriophages? They can attach, they have these cycles, so they are here attached to a cell and can undergo two different lifestyles. So one is they attach to the cell, insert their DNA, and then the DNA, this hijacks the bacterial replication system, produces more of these bacteriophages, and then eventually lyse, and you produce more phages. And so in this manner, though, they also kill the bacteria. The other important life cycle that they have is the lysogenic cycle. So here, the DNA is inserted into the bacteria. Then that inserts into the host gene, the DNA, and then that can actually induce changes in the host bacteria so that it can actually be more fit and actually perhaps replicate more. Or it could also induce antibiotic resistance genes. So there are some advantages and potential disadvantages of this happening. And so this is why we care, because the phage and bacteriophage are the same thing. These phages can impact human health. So the Shiga toxin and cholera toxin, for example, are from phages. They can also determine virulence and pathogenicity, can bring about antibiotic resistance, and actually change metabolic capacity. Oops. And I put this here to remind me the German outbreak about maybe close to 10 years, between 5 to 10 years ago, that was an E. coli, a Shiga-producing E. coli, Shiga toxin-producing E. coli that were decidedly from sprouts that affected nearly 4,000 people. That actually started with phage, and then it was transferred actually from one E. coli to another E. coli, so bacteria to bacteria transfer that then increased its pathogenicity and virulence. And in addition, there's even there's phages responsible for fluoroquinolone resistance to those same Shiga toxin-producing bacteria. So just an example of how these phages impact our life. So before I go into a little bit more, I just want to show how difficult it is to study the virome. This is the everything that's involved. When we have either stool or breast milk, there's a lot of filtering involved to filter out cells, cellular debris, to get rid of human DNA that's there. And so it's before the sequencing. So it's not quite as easy as studying the bacteria. And that's partly why there are so few studies out there, because of the difficulty and the challenges, and also because of the cost. So these are the studies that have looked at the virome. It's actually not a complete list, so it seems like a lot. Some of these, however, have been using PCR studying, using specific viral primers and primers for bacteriophages, while others have looked at sequencing. But you could see that it's really been looked at many different sites, very similar to the bacterial sites that was just presented. So oral, gut, blood, urine, CSF, that actually just came out earlier this month. Multiple studies in the respiratory tract, only a few studies in milk, a few studies in amniotic fluid, one of which showed that there was there were no significant viruses in the amniotic fluid, and then skin. And these are all the different types of viruses that are there. And so there are many, but what we'll soon get into a little bit more is that a lot of these are bacteriophages. So we decided to look at the milk virome. We wondered if it was different from other body sites. And so you could see here, this is the adult milk in blue, and infant feces in red here. And then they separate out from the adult urine, adult feces, and adult saliva. So they are it's a different community in the milk and in the infant gut compared to the other body sites. In addition, it seems like the milk and the infant gut tend to cluster closer together compared to the other adult body sites. And when we look at mother and infant stool together, mother, milk, and infant stool, a lot of them cluster together, but there are a couple that cluster a little bit separately. So we wondered, and then when we look at what was there, you could see here, these are the eukaryotic viruses, and then the bacteriophages, and over 95% were bacteriophages, both in the milk and the stool. And so we wondered if, and these are the top three there, the myoviridae, doesn't like me right now, pushing it too fast, okay. the myoviridae, cephaloviridea, and podoviridae, and they're really the differences are the tail of the phages. And then when we look at diversity, the viral diversity tends to correlate with the bacterial diversity. So stool, this is infant stool, and these are infants that are less than 1 month of age, so very young infants. The stool viral diversity here is actually still quite low, where milk viral diversity is higher. And this is the bacteria. Again, the milk is higher in diversity than the infant stool. And so this actually is similar to adult data, where they've looked, this was from a twin study, and they looked at bacteria diversity, and there were some where the twins had similar diversity, and some where they were discordant, shown here. And the viral, here on the x-axis is the microbiome, so the bacteria alpha diversity, and then here is the viral alpha diversity. And what we see is that as you increase the microbial biodiversity, you also increase the viral biodiversity. But interestingly, this is only seen with the bacteriophages and not with the eukaryotic viruses. So the bacteriophages, which infect the bacteria, go up in diversity with the number of bacteria, but the eukaryotic viruses are pretty stable without dependence on the microbiome diversity. So these phages, remember I talked a little bit earlier about the 3 most common phages that we saw, have these different tails, and that will determine how it attaches to the different bacteria and who it can attach to. So, for example, these are the different attachment, the specific bacterial receptors that are found in gram-positive organisms. And then here in the middle here, found in gram-negative, in both organisms, and then down here, gram-negatives. And so depending on the type of tail, it can look for certain receptors in order to be able to insert its DNA. And so this is important because that will determine what type of bacteria it can infect. So virome transmission factors. So there's a lot of different ways that viromes can be transmitted. And so there's some that are modifiable, or some that are fixed, and then others that are potentially modifiable. So age, sex, genetics, that's all fixed. But people have looked at differences in adult gut microbiome and also in blood, and these were all affected by age, sex, and genetics. And these are the potentially modifiable factors. So potentially, delivery method sometimes is modifiable. We're definitely seeing a lot more cesarean deliveries than we used to, and so some of that may be some people may have an elective C-section just because it's easier. But that also, in addition to changing the microbiome for the infant, that also changes the virome. The environment affects the viruses that you have on your body. Household contacts, there's been a lot of studies of people living, adults living together. They tend to share viruses for a period of time, then it goes away, and then somebody may bring in a new virus from the outside, and then they share it again, and it goes away. Antibiotic use also affects the virome, most likely because it affects the microbiome, and then therefore the bacteriophages might go down in number and diversity. And then diet, breast milk. So very early in life, the first thing that happens is birth, right? So as I talked about just a little bit earlier, in the during pregnancy, they didn't seem to find a lot of significant numbers of viruses in the amniotic fluid. So then the next thing that happens is birth. And whether you're delivered by vaginal or C-section, You could see this is the beta diversity of the babies born via C-section compared to vaginal, and they do cluster distinctly. And these are the different types of viruses that are there, and again, differ by C-section versus vaginal delivery. So that's an important factor. The next is breastfeeding. So this was actually shown earlier today, but just like we looked at in breastfeeding with bacteria, we saw that in bacteria, if you're exclusively breastfed during the first month of life, the moms and babies actually share about 35% of the microbiome, the bacteria microbiome. And then that decreases over time, which makes sense as the baby starts to feed from other means, the introduction of solid foods, and also gets around in their environment, crawling on the floor, putting things in their mouth. That changes the bacteria flora. And so we wondered the same for, and so we think that a lot of that is mom to baby, but there's probably a component of baby to mom, and it just sort of cycles between mom and baby. And so we asked the same question for viruses. So do the viruses come from mom's milk, or could it be the same as other moms, or just kind of random from the environment? And we know that viruses can come from milk. There were some hints earlier with the mom's milk, the communities being more similar to the baby's milk. We also know that there are viruses such as CMV or HIV that transfer from mom's milk to the baby and can infect the baby. But what we didn't know as much about are the bacteriophages, which comprise 95% of the viruses that are there. So this was the first study that in 2008 that looked at both phages and bacteria that transferred from milk to baby. And so they found that there were phages that infect lactobacillus, lactococcus, streptococcus, and enterococcus, species that were all dominant and abundant in milk. But they did not actually find the viral sequences of the phage in the milk itself. But the method using here was using primers against some bacteriophages of significance and not looking at all of the virome. So, and then this paper, which came out in 2017, looked at the maternal inheritance of bifidobacteria communities and bifidophages. So they focused in specifically on the bifidobacteria and their phages. And they actually showed that there was vertical transmission. So then they sequenced the bifidobacteria in milk and the bifidobacteria in the baby. They found that they were the same. And they found also that the phages were the same. And what they found, though, was that the phage was transmitted as a prophage. So in the bacteria, it had already integrated into the host gene. It was transmitted as a prophage, and then in the baby gut actually was able to replicate and become more phages, leading to cell lysis, but having lots more phages to go around to all of the other bifidobacteria. And then in our study, we also looked at homologs within pair. So again, mom and baby pairs together, how much of their sequences were the same compared to baby with a random mother pair. And so you could see if we, within pair, the numbers were always higher than if we paired that baby randomly with another mother. These were the viruses that we found in the three pairs that had the most homology. And I really put this up there just to show that there's a lot of homology, but also that the significant amount of what was shared were bacteriophages. So the question is then, once these phages and other viruses get to the gut, what happens to them? How long are they there? And unfortunately, there's not a lot of data about this in pediatrics, but in adult data, this was a study of one adult man grabbing stool at multiple time points. And they looked at how different those communities were by the interval between time points. So this is only 1 day, 10 days, 100 days, 850 days. And you could see, and so then the higher the number, the less divergent, and the lower the number, the more divergent. So here, if it was only a few days in between samples, you didn't see a lot of change. And then as you increase to the large number of days, 850 days, so 2.5 years, then we see more changes. And the colors, actually, I don't know how well the colors show up for you, but the temperate viruses in blue are the ones that are more often transmitted as prophages or are integrated into the bacterial gene. And you could see that there's less change for them compared to the not temperate viruses. And on your right-hand side, we look at the viral families and the substitution rate. So as the DNA replicates on itself, then occasionally there will be substitutions into that, and that could lead to new genes, new antibiotic resistance, et cetera. And so the microbiome had the highest amount of substitution. It was about 4% in this particular study. And that it was suggested that it's possible because that's a single-stranded DNA phage compared to the rest, which are double-stranded. And so with the single-stranded, it's a lot more error-prone compared to the double-stranded phages. And then phages then can move between individuals. Now, this study is a study of fecal transplant, so it's not at all the normal way that phages move between individuals. But I think it's still interesting. It's three pediatric patients who had ulcerative colitis, and they were treated by stool transplant actually between 22 and 32 times. So they received fecal microbial transplant 22 to 30 times, which seems like a lot for me. But anyway, it's from a single donor. And so on this side, on your left, you have the patient, their original virome, and then your donor virome just below them. And then the first column is pre-transplant, then during the transplant, and then post-transplant. And what you could see in pink here, a lot of the pink had the largest amount that stayed around post-transplant in all three of them, and that was the Virome, which is interesting to me because that was also one of the highest abundance for the phages in our studies when we looked at maternal milk going to the infant gut. The Virome was also one of the highest that transferred and then stuck around for a little while. So these are the only studies that actually looked at milk and infant gut samples over time. And what they found was that this is the bacteriophages, and then this is the eukaryotic viruses. So the phages actually start out quite high and then decrease over time. And then the eukaryotic viruses actually start low and increase over time. And that makes sense to us as pediatricians because the kids will encounter different viruses in their young life, and then some of those persist in the body over time, and so that has a slow, gradual increase over time. But these bacteriophages go down, and this study compared bacteriophages in orange here, and the bacteria in blue. And so you could see that the bacteria, as the bacteriophage goes up, the bacteria comes down. And same here, that basically there's this predator-prey relationship between the bacteriophage and the bacteria, and some people call it kill the winner. The predator-prey suggests that there's some cycling up and down of the different of the phage and the bacteria. So here is depicted here. We have the phage here kind of going down, but cycling a little bit, where the bacteria go up in number. And then in blue down here are the eukaryotic viruses. And this all tends to stabilize over time, similar to the bacteria, around 3 years of age. But this whole middle section with the dotted lines are really a question mark, because we don't have studies we don't have enough longitudinal studies with enough numbers to really know if this is true. This is all hypothesized based on the 2 studies that you saw earlier on what happens with the phage and the bacteria. And so one wonders, with this change over time, and with the number of bacteria going up and down, if any of those kind of cross these windows of opportunity, where there's some critical time periods and time points in early life where the microbes, particularly the bacteria, influence immune development and metabolic development. And so if the phages are there, then perhaps they're also helping to control the bacteria so that there's not too big of a proliferation of the pathogenic bacteria, or the ones that don't induce immune tolerance correctly. And it's interesting to me that Mother Nature has sort of introduced all these different factors perhaps to control the bacteria, the potential for IgA and an antibody in milk to bind to certain bacteria, potentially pathogenic bacteria, so that they're not misbehaving in the infant gut. There's the potential for oligosaccharides to act as prebiotics, to feed certain bacteria, to favor certain bacteria over others. And then there's the potential now for viruses also to either infect and kill off bacteria, or to introduce the good genes in them to make them more fit in their environment. And so this is one study that just came out recently that showed whole fecal metagenomes and viromes, where they looked at the potential for both bacteria and viruses to act, so different cellular functions that are possible. And so you could see up there some of the different processes, lots of cellular processes, metabolism, and then here specific KEGG pathways that are involved. And in there you see a lot of metabolism, infectious diseases, which also implies immunologic diseases or immunologic teaching, and many other processes. This is in adults, but still possibly transferable to pediatrics. It's something that needs to be studied. So now I'll present just the very few studies that have been done in pediatrics. So this is there was one group that looked at infants with diarrhea, and they classified them by the type of stool. So from pellet, so that's, I guess, maybe too dry, but maybe a good stool, all the way to watery, with several classifications in between. And so they looked specifically at the eukaryotic virome and saw that there were changes associated with the type of stool consistency. But it's really hard to know, was this the cause or the effect, or just an association, maybe neither, just an association. But these are so far, and you could see this was just published 2019, these are so far the only types of studies that are out there. Another study, a little bit out of order. Another study looked at autoimmune diseases and specifically type 1 diabetes, and found that the intestinal virome changes precede autoimmunity in type 1 diabetes in susceptible children. So again, hard to know cause and effect, really just starting with an association type of study. And then Jeff Gordon's group looked at malnutrition. So they looked at virome patterns in healthy and malnourished twin pairs from Malawi. And they found that virome patterns were concordant in healthy twin pairs. So if you have two healthy pairs, the viromes were very similar. But then if you had a one twin became malnourished, then you also saw some similarities. Actually, both viromes became altered, and there was delayed gut microbiome development associated with it. You all probably remember he had that their group has published extensively on the gut microbiome and showed that there's delayed gut microbiome development or maturity in children that are malnourished. And so what we saw there is that when they looked at the viruses, what they found was an increased prevalence of this virus family, Anelloviridae and Cercoviridae families and phage populations that could distinguish the malnourished twin from the healthy twin. And these two families have also been associated with increased diarrheal illnesses and also with looking at adult HIV infected versus not. The HIV infected also had an increased prevalence of this Anelloviridae family. And then another group in China followed around 4,500 children over a respiratory season, over several respiratory seasons, and looked at how many times they developed acute respiratory tract infections. And so what they found, they looked at all of the virome, but they focused in on this propionic bacteriophage, and they found that kids that developed multiple acute respiratory tract infections had higher amounts of this phage, significantly higher amounts of the propionic bacteriophage, compared to those that only had a single acute respiratory infection. And here you could see this, it goes up with the actual number of respiratory infection, first, second, third. And what they found was this propionic bacteria actually had the ability to induce certain increased amounts of cytokines in the respiratory tract, perhaps leading to more symptoms, more inflammation in those children with multiple respiratory infections. Oh, and then this study also just came out earlier this year. In Bangladesh, they looked at children who were vaccinated with polio vaccine, and then looked at immune response. So in the lighter color here, seronegative, in the darker color, seropositive, meaning that they converted and developed increased antibody. And they looked at the amount of the prevalence of other enteroviruses. So the question was, could other enteroviruses interfere with vaccine response to poliovirus? And what they saw was yes. So you could see in the seronegative group, again in the lighter color, they all had higher amounts of each of these enteroviruses in their gut at the time of vaccination, and they did not respond, and so they did not respond as well to the vaccine. So it's an interesting concept that these viruses can also interfere with antibody response and affect the immune system. So those are all the studies that were done in children. So again, not very many, but one can kind of think about, potential next steps and potential uses for viruses if we were to find them to be helpful. So here, kind of nicely documented in a figure, potential phage-inspired approaches. So you could give phage therapy where it, mostly attacks the pathogen, the pathogenic bacteria, and so then they all die off and you're left with some good bacteria. You could have phage enzymes, kind of similarly doing the same thing, biofilm dispersal by the same method, drug sensitization. So you can kind of introduce phages to either, again, promote the bacteria that are present or kill off the pathogenic bacteria that you don't want to be there. But there's still a ways to go. There's a lot of limitations of the current studies, and most of the studies that we know of so far are cross-sectional data with a small number of subjects. And even in the few that were longitudinal, again, a very small number of subjects where we have data over time, especially in early life where there are so many changes to the baby. And there's a lot of studies where they only look at the baby, but it doesn't include mother and infant as a pair, so we could actually study the milk microbes and viruses and the baby's microbe and viruses. And the technology is still expensive. It misses a lot of latent viruses, integrated viruses, double-stranded DNA viruses, and then large eukaryotic viruses. So there's still a lot of unknown. These actually, in several studies, can be up to 70, 80, even 90% of the viral particles that are there. So we really are just knocking at the very tip of the iceberg, and it wouldn't even be like the iceberg picture you showed earlier. It really would just be like a peck of the iceberg. And there's a lack of standardized protocols and incomplete viral databases. So that's the other part of the problem is that we use the viral databases that are out there, but because many of these viruses are novel and unknown, it's hard to tell exactly what they are. So I'll kind of leave you a little bit with the slide of what we hypothesize happens. Kind of a summary of some of the things that I talked about, that there are all of these different factors that influence the virome, both in mother's milk and in the infant gut. And there's this trajectory over time that's still unclear, where we need more data, but I think it's a nice potential field of study. And then just like with the bacteria, we need more mechanistic data of what actually happens to try to move beyond the association data. So with that, I'd like to thank, these are lots of students that work with me, my former mentor, some collaborators. We're extending our studies. So I am actually a pediatric infectious disease doctor and by training more of a vaccinologist. So I'm very interested in immune responses to vaccines. So we are looking at, and so there are some vaccines that have significantly different rates of response in the developed countries compared to developing countries. And so we're trying to understand why, kind of focus on the immunologic aspects, and then trying to understand how the viruses and the microbiome play a role in that. And so these are collaborators there. And then at UCSD, David Pride, who I worked with on our Virome studies, the Rick Bushman, University of Pennsylvania. When I first started on the microbiome, I went over there and spent just a couple days in his lab talking with the wet lab people and the bioinformatics people to kind of learn all of this. So thank you very much.
This is now open for discussion. Go ahead. Can I ask first? Yeah. Okay. Thank you very much for your nice presentation. As you have mentioned, a study from Bangladesh referring to the immunogenicity of polio virus. So we know, by and large, when a phage infects bacteria, so they multiply inside the bacteria, okay, and ultimately the bacteria dies, and the viral population increases. So by law, there should be more immune response if there is more bacteria which is infected by the polio virus, and the bacteria dies and the virus flourishes up. So there should be more immunogenicity, and antibodies should be more in those cases. I mean, I'm contradicting the thing you have presented. This is one thing. Second, I like to a second question. That is, you have mentioned that persistent latent viruses. So you have included human herpes virus type 6. We know the human herpes virus type 6 causes roseola or erythema infectiosum. So it's a disease of the babies between 6 months to 2 years of age. They have a high grade of fever for 4 to 5 days. Then suddenly fever goes by lysis and the rashes appears. When the rash appears, the baby becomes all right. And I do not know how these bacteria, why these bacteria remain latent or persistent, and how they present later life. Like your herpes simplex virus, type 1 and type 2, they hide in the somatic nerve endings, and the herpes zoster virus in the posterior root ganglia or in the cranial nerves ganglia, and they flourish up as shingles later on life. But how the herpes virus type 6, where they survives, and what is the late presentation if they are latent or persistent? And I don't know the herpes simplex virus type 7, what type of illness they cause. I do not know. If you know, you can acknowledge.
Yeah. Okay. So the two questions. So one, the first question on the viral shedding and the vaccine response. This study focused mostly on the eukaryotic viruses and the enteroviruses specifically. So not on the bacteriophages. And so it wasn't looking at phages that would infect the bacteria and then lyse them. So I think that...
So it differs on prokaryotic and eukaryotic virus?
Yes, differences between eukaryotic viruses and bacteriophages. Yes.
Okay.
So it's the bacteriophages that will affect the different bacteria that could either cause better fitness or lysis. And so I think that's what you're thinking about, that potentially if there's lysis, perhaps there would be a bigger immune response. But in here, it was that there were enteroviruses there, perhaps that may look somewhat similar to poliovirus, and so therefore they're vaccinating with poliovirus, and the immune system then thinks it already knows, how to respond to that particular virus, and so doesn't mount an immune response. Yeah. So, and then your second question about HHV6, so most of the time for us, it doesn't cause lots of problems later in life. When we see it is if you have somebody who's immunodepressed later in life, then the HHV6 will come. So in those with HIV, those who are immunocompromised from chemotherapy or mainly bone marrow transplant, so those types of patients.
Where they lie dormant? In which part of the body?
That's a good question. I don't know the answer to that. I don't know if any audience members know. Yeah. Yeah.
Sure. There are 3 viruses in humans of enormous medical importance associated with milk. HIV you mentioned briefly, and I would like to add that, of course, you can be infected that route, but it's relatively very rare. Mm-hmm. And as a matter of fact, WHO recommends for mothers who are HIV infected to continue breastfeeding because the probability of being infected with HIV is much lower than dying of diarrheal disease. Second, EBV. Epstein-Barr virus is present in human milk in relatively high proportion, and interestingly, children in Africa within 1 year of their life are all EBV infected. 96% of kids are seropositive for EBV, and apparently it comes from milk. This is not the case in Western Europe, where children are infected around time of adolescence, and in China, EBV infection 6 to 8 years. And again, there is a tremendous impact on that. I don't know how many of you are from Africa, if anybody is from Africa, but EBV, as you well know, causes infectious mononucleosis. Yes. It's a disease which is unknown in Africa, due to the fact that kids are infected so early in their life and they mount without basically any perceptible clinical manifestation. They mount an immune response for lifetime and they cannot be infected with EBV by the oral route and have infectious mononucleosis. And maybe you can say something else about cytomegalovirus. Again, tremendously important virus, which goes before the birth or through the milk, and you know the consequences. So I think these are three viruses which really should be always reminded of us milk, because that's the way how they're transmitted.
Yes, thank you. Yeah, thank you for bringing up those very important points. I didn't choose to concentrate on those viruses because there's actually a lot more known about them, but I think I just want to reiterate your important point about HIV, that it can be transferred through milk, but is rare, and so the recommendation is to continue breastfeeding exclusively for 6 months and to a year if possible, and then to have a slow wean because they have found that if you wean rapidly, you actually have a higher risk of transmitting HIV from mother to infant. And then CMV, I know there's several neonatologists here in the audience, and so as you well know, there's lots of consequences potentially of CMV, particularly transferred in utero, but possibly also transferred to a premature infant. So yes, thank you for bringing that up.
Yes.
Hi, thank you for the wonderful talk. Vanessa Castagna from UC Davis. So I wonder if you could comment on bacteriophage therapy. This is known to be successful in eliminating bacterial infections. And what are your thoughts on utilizing bacteriophage therapy in modulating the microbiota in perhaps a dysbiotic state?
Yeah, that's a good question. I think that eventually that would be a nice therapeutic method. I think right now I feel like we don't know enough yet because there are so many different bacteriophages there, and we know that there are certain ones that are predisposed to infect certain bacteria, but it's hard to know if they also can potentially infect other bacteria, and so that perhaps you'd be doing a disservice if you're killing off good bacteria, killing off both good bacteria and bad bacteria. But I do see it as a potentially promising therapeutic in the future.
Yes.
Thank you very much. Nalini Jongriyapan, Mahidol University, Thailand. Thank you very much for your wonderful talk. You mentioned about diet, breast milk, and we all know that maternal diet affect nutrients in breast milk, and as well including prebiotics, right? That can affect probiotics or bacteria in infant gut. What about the effect of maternal diet on breast milk with virus?
Yeah, I think that's a great question, but there's no answer that's known at this time. There just isn't a lot of data, or there isn't any data that I know of at this time to look at maternal diet and which viruses are in milk and how that affects the infant gut. But I imagine that data will come out soon as there is, more, as there are more people looking at the virome and learning from the past now, grabbing epidemiologic data as well. So hopefully we'll know soon.
Pia, there's a lot of parallels between bacteria and viruses. What happens when women are given antiviral medication? Does that disrupt the virus and its association with bacteria?
Yeah, that's a great question. I don't think there's any data out there for us to know for sure. Now, there are several antivirals that are out there, but they don't one, they don't work very well. And two, there's very few. So acyclovir works very well for HSV, but not actually so well for maybe the rest of the herpes virus family. And not so well for other viruses. And same with the other antivirals that exist. And most of the time, they're not prescribed for routine viral illnesses. The only ones that are prescribed for routine illnesses would be acyclovir for herpes virus or oseltamivir for the flu virus. So I think we don't know yet what effect that might have on the other viruses, but I think maybe not as much as we might think, unlike antibiotics, just because our antivirals are not that effective in general. Yeah.
I have another question with regard to the Jeff Gordon studies, where they showed that children who are malnourished have an immature bacteria that persists to later in life. Does that same thing happen to viruses?
Yeah. So in this study, they looked at development over time. So it looked like the viruses also were delayed, but it looked like that the delay was mostly because the bacteria were delayed in development.
So it's dependent on the bacteria.
Yes.
Okay, thank you. Are there any other questions, Ray?
I'd like to ask one question. Last one is that you have mentioned that 1 ml of seawater contains 10 to the power 7 viruses. And we often go to the sea beach and swim in the sea water. So is there a chance of having viral infection from the sea water?
Well, a lot of the viruses in the seawater are, a lot of bacteria. How do they survive
in the increasing salinity in the seawater?
Yeah, there are a lot of phages and a lot of, all of the different type of viruses, but the majority are phages. But humans swim in the ocean all the time, and so I think the risk of being infected there would be just like the risk of being in the environment, and probably the viruses that infect us most we get from other humans. So contact with other humans will give us those viruses.
Are there any other questions? If not, thank you very much.